Hello students. In this video lecture we're going to learn about animal diversity. So I have here a list of 19 important lineages that we have identified across the animals. So you can see things like sponges and cnidarians and flatworms and annelids and mollusks and tunicates and echinoderms and monotremes and marsupials and placentals. And maybe some of these words seem familiar to you. You might quickly look and say, maybe half. And then maybe there's others that don't seem familiar at all. And so our objective today is to familiarize ourselves a little bit with some of these groups, learn how you could familiarize yourself with the rest of the groups, put these groups into a matrix where we identify morphological characters and whether these characters are present or absent in these groups, and then use that matrix to reconstruct a phylogeny, just as we learned in previous lessons of the animal groups. Okay, so the three that we're going to focus on for right now are cnidarians, insects, and birds. So cnidarians are these organisms that live in the oceans, and they also can live in fresh water, but they have radial symmetry, which means that you could take them, and if you turn them right up onto you, you could slice them in any direction just like a pie, and you get equivalent parts on all sides, right? They also are the first organisms to develop these true tissues where they have an ectoderm, an endoderm, and this mesoderm layer, so in yellow here. So you have essentially these different kinds of cells that start to specialize to do certain functions, and they form tissues, right? Because you get lots of cells together and you form a tissue. So you get these really these first simple animal tissues begin to develop, and cnidarians have these. Cnidarians also do not possess a complete digestive tract, so they don't have a mouth and then going all the way down the body and with waste coming out of an anus. Rather, they have only a mouth-anus opening. So for example, in this Medusa form here, this jellyfish, right, the food would go into the mouth, it would be digested as much as possible, and then the food would come out of the same opening that it went in. Cnidarians are also unique in that they have these stinging cells. So these are these nematocysts, or these cells that have these little barbs, and they have a sting venom associated to them. And cnidarians also can reproduce either sexually or asexually. So that's a quick overview. And again, a lot of this information you could just go to Wikipedia or go to any online search and find a lot of this. The next group that we're going to highlight are the insects. So the insects are arthropods, which means they have hardened exoskeleton and that the legs and appendages are jointed. And insects have 6 jointed legs. They have 3 pairs of legs. Their body is also divided into 3 parts: the head, thorax, and the abdomen. All insects have 1 pair of antennae coming off the front part of their head, as you can see in this leaf beetle here, or in the butterfly up here. Insects also are the most diverse group of animals. That we know this because, first of all, we've described more insects than anything else. More than 1 million insects have actually been written down with a name and been described. And that's a lot of organisms. Also, insects we know that are the most plentiful and diverse of all of the animals because we haven't described a lot of the diversity that's out there. There's millions more of species that we haven't described yet. Insects, in order to grow, must molt that exoskeleton, that outer covering, in order to get bigger each time. Just a quick clarification here. Insects only molt that outer covering when they are going through their immature stages, so larvae stages or nymph stages, up through the pupa, and then they will go through their last molt to become the adults. So all of the insects that are figured in the diagram there are all adults. They're never going to get any bigger than that. That's as big as they get. Once they get to the adult stage, they don't go through any subsequent moltings. Whereas things like crabs can kind of continually do that their entire life. They can get bigger and bigger and bigger and so forth. So I just wanted to clarify that one little point. Insects also have bilateral symmetry, which means that instead of radial symmetry like before where you could slice them like a pie, they have a left and a right side. Insects also have a protostome, or they come from a protostome-like development. So what this means is in the early dividing of the cells, once the zygote forms, right, the egg and the sperm come together and forms the zygote, and then the cells begin to divide. Initially, you get particular types of dividing called cleavage. You have spiral cleavage and radial cleavage and so forth. But what we're interested in is once these cells start to actually take form and they invaginate, they turn inside, they start to turn in on themselves and form this indentation, that very first hole here, which is called the blastopore, becomes the mouth. And that's how it is in protostomes. In deuterostomes, the first invagination becomes the anus. And so that's a big difference that can discriminate between organisms that had an evolutionary history as a protostome or an evolutionary history as a deuterostome. So that's a quick overview of insects. So now we're going to look at birds. Birds are endothermic organisms. They have 4 limbs. Now, a lot of times you say, wait, they've only got 2 legs, but they've got the wings. The wings are the same as the 4 limbs of all other organisms. They have many flight adaptations, like feathers, bones that are hollowed out and have air sacs within them so that they're very light. They have lots of other, aerodynamic adaptations as well to allow for flight. Birds are the only surviving lineage of the dinosaurs. So when someone says the dinosaurs are extinct, that's actually false, because one lineage did survive, the birds. Birds are also most closely related to things like crocodiles and alligators. They possess true amniotic membrane with a hard egg shell. So if we look at this diagram of the egg over here, here's the embryo or the growing bird. But around this, there is this membrane. This amniotic membrane, or the amnion, protects the growing embryo, but it also prohibits it from drying out. It can cushion it as it's inside the egg and so forth. It's a really important membrane for the development of true eggs. Birds, of course, also have the hard shell on the outside and the yolk sac and this other structure over here that's mostly for waste and gas exchange called the allantois. Now, birds also ancestrally have teeth set in sockets. So you think of a bird and all they have is the beak, but actually, developmentally, you can actually see this in birds when they're in embryo. You can see their sockets, you can also see the teeth starting to grow, but then they're reabsorbed and then teeth are not there. Interestingly, though, there are many labs around the country that have figured out how to go in and tweak a developmental gene, and birds can grow with teeth. So birds have teeth. They just don't have them in the adult form. Birds also have antorbital and mandibulate fenestrae. Now, what these are openings. The antorbital is an opening right in front of the eye, so it's a hole in the skull, right, that's in front of the eye. And they also have a hole in the jaw, right, around here. They're not as profound in modern-day birds, but in the fossil record, they're much easier to see as well. And those are defining characteristics that unite birds with other organisms as well, these openings in front of the eye and the openings in the jaw. So there's a quick review, right, of these 3 organisms, 3 groups that you also could have learned a lot about just doing a quick Wikipedia search. So if we were to do this for lots of the organisms, you can start to accumulate what's called a morphology matrix, where you have these zeros and ones, just like you have learned before in some of the other lectures. And you can start to code which of these characters are present or absent in all of these 19 lineages that we've identified. So for example, in the Cnidarians, we could fill in the multiple cells, the true animal tissues, and the radial symmetry with 1, because that's what they have. And in insects, they also have multiple cells. Remember, they were a protostome, not a deuterostome, and so we would put the 1 was over here, and we put a 0 in the deuterostome. And insects also molt their outer skeleton when they need to grow, and they also have antennae. So we could mark both of those. And then in birds, they're multicellular. They also are deuterostomes, not protostomes. Birds have tetrapody, which means that they have 4 limbs. And birds do not have the 1 temporal fenestra. They have the 2 temporal fenestra. So it's just that the openings in the skulls that we're talking about here. So you can, again, fill this out for all the characters, and sometimes it's easier to maybe even work your way down a character. So for example, if we looked at antenna, we know that archaea don't have antenna, sponges don't have antenna, cnidarians don't, flatworms don't. Annelids actually do have a form of antenna, but it's not the kind of antenna that we're actually talking about here. So sometimes it requires you to really define what the character is here. Mollusks don't, roundworms don't, crustaceans do. So the only 2 organisms in this entire row of antennae that would have a 1 would be crustaceans and insects, right? So you can work both down columns and across columns, but eventually you fill out your entire matrix with all 0s and 1s, or whatever it might be. If you're looking at genetic characteristics, this could be nucleotides, A's, G's, C's, and T's. So you fill this all out, and then what I think makes it easy to interpret and to figure out what the tree is, the phylogeny, is maybe to color in or somehow identify all of the ones being separate from the zeros, so that when I look at this now, I can say, oh, okay, the character multiple cells is saying that all the organisms, right, if you were to draw like a circle around this, all organisms except for archaeal bacteria have multiple cells. And then we move our way down, all organisms except for sponges and the bacteria have true animal tissues. And only cnidarians and echinoderms have radial symmetry in the adult stage. And then we come to the bilateral symmetry. Well, everything has bilateral symmetry except for cnidarians, sponges, and archaea. And the reason we have it down here for echinoderms is because as larvae, they actually are bilateral. They have bilateral symmetry. It's only they kind of secondarily revert back to a radial symmetry in the adult, and so forth. And so you can keep doing that. And each one of these characters essentially becomes evidence for groupings. So maybe the easiest way might be actually to start way over on the far side with the marsupials and placentas. They share a characteristic called theria, which means they give birth to live young. They share that in common. So perhaps it's easiest to say, okay, we ought to put marsupials and placentals together on the tree. Then we ought to maybe add the monotremes to those 2. And then we ought to start a different grouping over here where we have the crocodiles and birds. Here's another characteristic that just also unites the monotremes, marsupials, and placentals. And here's a character that now unites the lizards and snakes with the crocodiles and birds, and so forth. And we just keep working our way back, and eventually you'll make a tree, right? So coming back, here we are with the marsupials and placentals, the marsupials and the placentals with the monotremes. Here with that crocodile and bird lineage, we added the lizards and snakes, and then we had to add all of those together. And then you added the amphibians, the bony fish, the sharks and rays, the tunicates, the echinoderms, and these are all of the deuterostomes. And then over here we have the protostomes, and so forth. And so this is how you build the tree. You use those characters to build your tree. And once we have that, we can also then go back and say, well, evolution is occurring across this tree, just like our stamping stations from the clade race. And we can assign these evolutionary events with the names of the characters. So we know that some point after bacteria evolved, then you had things like a nucleus, a mitochondria, and multiple cells evolving. Then you had things like true animal tissues evolving, and you had things like bilateral symmetry, and then you had things like, a bony skeleton, and the hair, and the placenta, and so forth. So you have all of these different characters that can evolve across this tree. And that's kind of a quick overview to both how you can learn about groups and try to figure out some of the key characteristics of groups, and then use that information in a matrix to eventually make a tree. Now, both of these, the matrix and the tree, you'll be using in some of your other assignments, so make sure and complete these and have a pretty good understanding about what's going on, but you can refer back to these, of course, as you work on your assignments and quizzes. So that's the end of our animal diversity lecture.